So we talked about what we were doing today in the lecture. I'm just mentioning this again for the recording. I wasn't recording. And I talked that we're starting chapter 4. Let me start sharing the screen. When I'm talking like that, and I was showing you the screen, I was showing you the syllabus, you got to tell me I'm not showing the screen. So um, I thought I was showing the screen. All right, we don't need that, so let's minimize that, and I can close that. So, Chapter 4, Functional Anatomy of Prokaryotic and Eukaryotic Cells. This slide is showing you the major goals, not the entire goals, just the major goals for this chapter, and a rough outline. So please know the terms. You should know what a coccus is, a bacillus, a spirilla, a strepto like streptococci, staphylo like staphylococci, diplo like diplococci, tetrad, a sarxinia, a vibrio, a spirochete. You should know what glycocalyx or glycocalyx is. You should know what flagella is. You should know what axial filaments are. Fimbriae and pili, peptidoglycan, endotoxin, selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid, inclusions, endospores, and lastly the endosymbiotic theory. And we'll talk about all of these topics in this lesson. About 2/3 of them we should discuss today. You should also be able to discuss how gram-positive and gram-negative cells walls differ and how that gives rise to the gram stain. We've already talked about that last part, but we'll hit it again and go into greater detail how the cell walls differ. And then lastly, describe the importance of taxis in bacteria. Any questions about what we're covering? All right. So you should know the differences between prokaryotic cells and eukaryotes. The Greek word for prokaryote is actually meaning pre-nucleus, strictly speaking pre-kernel, but we'll use the word nucleus, so before nucleus. Eukaryotes, true, meaning true nucleus. The prokaryotes have generally one circular chromosome, and the chromosome is not in a nucleus. The eukaryotes have linear chromosomes, and they are in a nucleus. The DNA of prokaryotes, does anyone know, are they associated with histones or not?
No, they're not.
Yeah, the DNA of bacteria are not associated with histones. That was sort of a trick question. The DNA of archaebacteria do have histones, but I'm never going to ask you about that. So you only need to know that the DNA of bacteria do not have histones. The DNA of eukaryotes is associated with histones. And if you don't know what histones are, it's just a protein that the DNA wraps around in the chromosome of eukaryotic cells. Prokaryotes do not have organelles, and when I use that term, I mean a membrane-bound organelle. And then eukaryotes have organelles, specifically membrane-bound organelles. And the nucleus is just one of the cell's organelles. The bacteria have peptidoglycan in their cell walls. The archaebacteria do not. So this is one time where once again you have to remember that this is applying only to the bacteria. And then the eukaryotes do not have peptidoglycan in their cell walls. When we're talking about prokaryotic cells, generally most of them divide by binary fission. That literally means the cell splits into two. We'll talk about that a little bit more in a future lesson. In eukaryotes, they generally divide by mitosis. And that we're not going to talk about You did cover it in Biology 160. Any question about any of that? So our next slide's an overview of what we're going to be doing. We're going to be discussing first the prokaryotes, discussing their cell shapes and their arrangements. That's a growth arrangement. The cell structures, the different parts within the cells, and then the movement of materials across membranes. After that, we'll then, if we have time, talk about the eukaryotes, talking mainly about their cell structures, largely the organelles, but we'll also talk about a few other cell structures. And then lastly, and if we don't finish that, like I said, I'll just assign it. It's in a, what do you call it, a worksheet, and you've had this material before. And lastly, I'll talk about the endosymbiotic theory. So when we're talking about prokaryotic cells, you should know their cell shapes and their cell sizes. The average size, at least on this slide, is between 1 and 10 micrometers. And really it depends who you're asking what the average size is, because most bacteria are between 1 and about 3 micrometers. But there are some that are longer and they can get up to 10 micrometers, at least in their length. They have different shapes. There are coccus cells, and they're a round ball or spheres. There's bacillus, and they are rods. And then there are spirals or spirilla, and they are a corkscrew type shape. And this is the start of the in-class assignment. So the rest of this material you'll be working on the worksheet, and so I'm going to skip over it. If you have any questions, you should ask me. Different types of spirals. There are some unusual shaped cells, and really you should just know the term pleomorphic. You should know about cellular growth arrangements, how the cells grow, and I think we talked about that in the lab. I'm just moving forward to where the worksheet ends. Okay, so the worksheet for this chapter ends around this slide. You should also know that some bacteria have a special structure called the sex pili, and it is a long, structure coming out of this cell here, and it literally skewers a F- cell, F positive cell, and we'll talk more about that later, and then can act like a winch and bring the two cells closer together. The purpose of the sex pili is to allow plasmid, the F plasmid in this cell, to move from this cell into that cell. And so the sex pili is used to transfer DNA from one cell, this cell, to that cell. And generally only a plasmid moves from this cell to that cell. You should know about the prokaryotic cell wall. It's a complex, semi-rigid structure responsible for the shape of the cell. Why the bacteria has a specific shape is dependent on the cell wall it has. And the cell wall also protects the cell and prevents osmotic lysis. So if a bacteria cell falls into water, it will expand the cell membrane up into the point where the cell membrane touches the cell wall. After that point, the cell wall prevents the cell membrane from exposing, and the cell membrane is expanding because water is moving from outside the cell inside the cell, and so the cell membrane will expand, but it can't expand any further once the cell membrane reaches the cell wall, and then that'll stop water from moving into the cell, and that allows the cell to not lyse, so the cell wall can prevent osmotic lysis, and the cell wall is rigid and then helps protect the cell, just sort of like being a structure around the cell. The cell wall can contribute to the pathogenicity of some species, where on the cell wall there are molecules which contribute to the pathogenicity of that bacteria, and we'll talk a little bit about that in a future lesson. In bacteria, the cell wall is mainly made up of peptidoglycan, and we'll talk a little bit more about peptidoglycan in just a minute. Any questions about any of that? No questions, move on. So peptidoglycan is a molecule found in prokaryotic cells, specifically the bacteria. It is a polymer of a repeating disaccharide. The two saccharides are NAG and NAM. NAG stands for N-acetylglucosamine. I said that right. NAG is N-acetylglucosamine. And NAM is N-acetylmuramic acid. I don't care which way you know them, you should just know NAG and NAM, or N-acetylglucosamine and N-acetylmuramic acid. And you just have a chain of these two disaccharides, NAG, NAM, NAG, NAM, NAG, repeating, and then the chain will be have another chain next to it, next to it, next to it, and then this plane will have a plane of NAG and NAM chains underneath it, and another one underneath it, and then one above it. And do you remember when I told you when you're looking at the gram-negative cells and it only showed you one peptidoglycan thing like that? Well, this is just a simple structure showing you the cell wall. In reality, it's like this, okay? And so even in the gram-negative, there's many layers, or at least several layers, I should say it that way, in the gram-negative cell. But it's much thinner peptidoglycan in the gram-negative cells than in the gram-positive cells. Any question about any of that? It's called peptidoglycan because NAG and NAM are saccharides and those are glycans. And then you see the peptides, short peptide chains, they link together, hooking this chain to that chain, meaning there's a short peptide running down from this NAM, and then it links to this short peptide going across, which then links to that peptide running down. So the peptides, sorry I moved that, there's peptides in peptidoglycan, and that's where the molecule gets its name. Peptides and glycans for peptidoglycan. Any question about any of that? Penicillin, that antibiotic, actually inhibits the lattice formation of the peptidoglycan, meaning see this link running across there and that link running across there. The penicillin prevents that link from forming. So when the peptidoglycan is being made, the cell wall is being put down, the penicillin prevents this link here, and that creates a hole in the cell wall. I'm trying to show you holes with my fingers. So this is peptidoglycan, and normally it has cross-links and all sorts of things. And there's a hole right there, and out of that hole the cell membrane can protrude. And as more water comes into the cell membrane expands out of that hole until the cell membrane eventually bursts through that hole in the cell wall. And that's how penicillin and other antibiotics that work in a similar way damage the cell. Any question about any of that? So when we're looking at the gram-positive cells, I think we've looked at this slide before. There's a gram-positive cell, and you notice that the yellow is the cell membrane, and it has a very thick peptidoglycan layer on top of the cell membrane. And here we're seeing the thick peptidoglycan layer. There are a few other molecules in here besides peptidoglycan, namely teichoic acid and lipoteichoic acid. Lipoteichoic acid simply has a lipid molecule attached to the teichoic acid, and the lipoteichoic acid runs to, the lipid portion runs to and binds to the cell membrane. The teichoic acid only runs through the cell wall. And both of these molecules help hold the peptidoglycan together. Any question about any of that? Oh, I already mentioned that. The teichoic acid may also help regulate the movement of cations through the cell wall. The cations can sort of use the teichoic acid to move down through the cell wall. And it's not essential because cations are lipid soluble, and lipid soluble molecules can move right through the cell wall because the cell wall is mostly peptidoglycan, and it is a polar molecule. It can dissolve in water. So water soluble molecules can move right through the cell wall. The polysaccharides of the cell wall can provide antigenic variation, and you can use the antigenic variation to classify certain cells. And like with E. coli, they have O157, H11. The O157 and H11 are antigens on the cell. And O157, for example, is an antigen on the cell wall. I think the H11 is also. Any question about any of that? The gram-negative cell walls are a little different in their structure. Once again, the yellow in the lower portion is the cell membrane, and the purple is the peptidoglycan, and the cell wall has a yellow portion above that. That's the lipids of the outer membrane of the cell wall, and that cell wall is much different. It's much thinner in its peptidoglycan, and it's more than just one layer of peptidoglycan, but it's fewer layers of peptidoglycan. And then it has an outer membrane, which is similar to the cell membrane, but different. The lower bilayer of the outer membrane is mainly phospholipids. It does have other molecules in it, mainly proteins, like the porin protein. But the outer bilayer of the outer membrane of the cell wall is different because, yes, it does have phospholipids, all the yellow molecules in here, but it has the blue molecules, which contain lipid A, and attached to that lipid A is an O polysaccharide, and so this molecule is a lipopolysaccharide. Lipid A is important because it's an endotoxin, and that means all gram-negative cells that have a cell wall have an endotoxin. Any question about any of that? Endotoxins are toxins, and toxins are toxic to us, so any exposure to a toxin is generally a bad thing. Let's look at the gram-negative outer membrane in a little more detail. It does have phospholipids. In fact, the lower bilayer, that's mostly phospholipids. And there are phospholipids in the outer bilayer, but it also has lipid A. And there are other molecules. The main one that we need to talk about is the porin molecule. And why that's important is that the cell does want things to come into the cell and leave the cell. And this outer membrane serves as a gatekeeper. And that is that if there was no porin molecule, then the only molecules that could move across the outer membrane would be lipid-soluble molecules. And obviously, cells want such things as amino acids and glucose and nucleic acids, or nucleosides, I should say, to move into the cell. And how they do that is they move through this porin protein. The porin protein is not a transport protein, which is generally how we move things through the cell membrane. If you want something to come into the cell and it can't move through the cell membrane, the cell will make a transport protein to move that molecule into the cell. Well, the porin protein doesn't move only one molecule. It's a big channel and it allows many things, but not everything, to get through. One thing that cannot move through the porin protein is penicillin G, the antibiotic penicillin G. But amino acids and glucose and nucleotides, they can move through this porin protein, and then they come to the cell membrane, because they can move right through the peptidoglycan. They're all water-soluble molecules. And then they have to move through the cell membrane through a transport protein. And then there will be one transport protein, generally speaking, one transport protein for each molecule you want to move across the cell membrane. Something you'll notice about the gram-negative outer membrane is it has no teichoic acid or lipoteichoic acid. It does have lipoproteins and then proteins in the outer membrane. The lipoprotein typically runs from the outer membrane through the peptidoglycan and then attaches to the cell membrane. All right, any questions about gram-negative and gram-positive cells? Let's see if I covered everything. The periplasm is the space between the outer membrane and the plasma membrane of a gram-negative cell. So this region here, which includes the peptidoglycan, is the periplasm. The gram-negative outer membrane can do other things for the gram-negative cell besides protection from antibiotics. It can protect the cell from the complement proteins. It can protect the cell from phagocytes, or help protect the cell, I should say. And it can have the O polysaccharide on it, which will serve as an antigen, like I mentioned E. coli. Although I don't think I said 157, but here's E. coli 0157, 87. That O polysaccharide is on the gram-negative outer membrane, giving this E. coli antigenic variation. We talked about lipid A and porin proteins. Any questions? If not, let's talk about how the gram-positive cell wall differs from the gram-negative cell wall. You already know gram-positive cell walls have a thick peptidoglycan layer. The gram-negatives a thin peptidoglycan layer. The gram-positive cell has got a little more rigidity to it than the gram-negative cell, but the cell wall is rigid, especially in comparison to the cell membrane, even in a gram-negative cell. The gram-positive cells have teichoic acid. The gram-negative cells no teichoic acid. The gram-positive cell no outer membrane of the cell wall. And the gram-negative cell has an outer membrane to the cell wall, and it does contain lipid A in that outer membrane of the cell wall. Any questions about any of that? All right, talking about the gram stain and how the gram-positive cell and gram-negative cells differ. The crystal violet iodine complex, when it gets in the, in all the cells, it will stain all the cells. In the gram-positive cell, that crystal violet iodine complex does not leave the thick peptidoglycan if the gram stain is performed correctly, meaning you apply the decolorizing agent for the proper amount of time. In the gram-negative cell, the decolorizing agent, the alcohol in it, dissolves the outer membrane of the gram-negative cell, and that's because this is lipid soluble, and so it will be damaged by the alcohol. Actually, even the acetone will damage it. And it'll just dissolve in the alcohol. That's why we say it will destroy the outer membrane. The crystal violet and the iodine will then wash out of the thin peptidoglycan layer, and it's just that it's too thin to hold on to the crystal violet. The safranin is then used to stain the gram-negative cells pink. All right, this slide is showing you some differences and a summary between the gram-positive cells and the gram-negative cells, and it is a good summary. You can ignore all the more than lower half of the cell. Peptidoglycan layer, thick in the gram-positive, thin in the gram-negative. Teichoic acid present in gram-positive, absent in gram-negative. Periplasmic space absent in gram-positive, present in gram-negative. The outer membrane absent in gram-positives, present in gram-negatives. The lipid A, the lipopolysaccharide, not present in the gram-positive cells, and you find it in abundance in the gram-negative cells. Now, with that stated, let me mention that there are some cells which are atypical from the gram-positive cells and the gram-negative cells. And so let's talk a little bit about that. I'm going to skip down to the archaeobacteria first because we're not really going to talk much about them. And they have a different cell wall than the bacteria. Most of the archaea do have a cell wall, but it's not based upon peptidoglycan. The archaea, generally speaking, don't have peptidoglycan in their cell wall. They have a pseudomurine, which is another disaccharide, to make the cell wall. And there's a few archaeobacteria that don't even have cell walls. And so these are atypical cell walls. What's interesting is the archaea, even though they don't have peptidoglycan, they do stain gram-negatively and gram-positively. Okay? And generally, the thicker cells, thicker cell wall cells are the ones that stain gram-positive. The thinner cell walls stain gram-negative. So it's very similar in the bacteria and their staining characteristics and why. It's just that it's not peptidoglycan that's making the cell wall thick and thin. Another atypical cell wall, and this is bacteria, so both of these two are bacteria, the mycoplasmas and the mycobacterium. The mycoplasmas are atypical because these bacteria lack cell walls. And that makes them a different shape and pleomorphic because they have no cell walls to give the cell shape. What they do is they have a cell membrane, much like animal cells, and they have sterols in their plasma membrane to help give the plasma membrane strength. And that's similar to the way animal cells have cholesterol in their plasma membrane to give it strength. All right, we're not really going to talk much about the mycoplasmas, but you should realize there is one family of bacteria that lacks cell walls, and these are mycoplasmas. Any question about any of that? We'll talk about the mycoplasmas briefly in one future lesson, I think in just one slide. Another atypical cell wall of the bacteria are the mycobacterium, and they are atypical because they're acid-fast staining cells. They have mycolic acid in their cell wall, and they have a different structure. And I don't have that structure here, but besides having peptidoglycan, they have mycolic acid in their cell wall, and mycolic acid is a lipid. And because there's so much lipid in the cell wall of the mycobacterium, these cells do not readily take up water-soluble dyes, such as the dyes used in the Gram stain. All right. Any question about the mycobacterium? We'll talk about them a little further in a future lesson. So there are 3 atypical cell walls, 2 of them in the bacteria, and they're generally in families. The mycoplasmas, it's a family of bacteria, and then the mycobacterium, another family. They just have different cell walls, we'll word it that way. Any question about any of that? All right, you can damage prokaryotic cells, and I'm not going to ask you to know the term protoplast and spheroplast, but to damage the prokaryotic cells, you can then make the cells susceptible to osmotic lysis. And that is exactly what penicillin, especially penicillin G, the antibiotic, does. It inhibits the peptide bridges forming in the peptidoglycan, and then that'll make a hole in the cell wall, making the cell susceptible to osmotic lysis. As more and more water comes into the cell membrane will expand, coming out of that hole in the cell wall. And then that will lead to osmotic lysis because the water will expand or come into the cell, and the cell membrane will expand until it comes to a point where it bursts. Another way to damage prokaryotic cell is with the enzyme lysozyme, and this enzyme digests the disaccharide in peptidoglycan, meaning it's digesting the NAG and the NAM linkage in the peptidoglycan. If you're not familiar with lysozyme, it's an enzyme you can find in your skin, and it's generally made by humans to help defend them against bacterial infection. Because when lysozyme gets on the cell wall to a cell, it can digest the peptidoglycan, making the cell then susceptible to osmotic lysis. All right, any question about any of that? I'm surprised because that was a pretty complicated slide. The gram-negative bacteria generally are not damaged as easily as the gram-positive cell, and that's because they have an outer membrane which can help protect the gram-negative cell. And one example is penicillin G. It works on gram-positives because it can get through the cell wall and then damage the peptidoglycan. But penicillin G cannot get through the gram-negative outer membrane and then damage the cells. And you would have to use another antibiotic, one that gets through the outer membrane of the gram-negative cell. Streptomycin, that's the one I was trying to remember last time. I was saying there's an antibiotic which can move through the outer membrane of the cell wall. Streptomycin is one. Streptomycin is also an antibiotic which can move through mycolic acid because streptomycin is a lipid-soluble molecule. There are, oh, it's right here, streptomycin. So there are antibiotics that work on gram-negative bacteria, such as streptomycin. But there are many family members of the penicillin family which also work on gram-negative bacteria. For example, ampicillin, which is one of the early members of the penicillin family, is not the first two to be identified, but it might have been the third member of the family. And it works on both gram-negative and gram-positive cells. All right, any questions about this slide? If not, let's talk a little bit about the plasma membrane. It is a phospholipid bilayer, and its main molecule is the phospholipid. And in the plasma membrane, it is mainly made up of two layers of phospholipids, and the phospholipids orient themselves this way, with the heads facing inside the cell, because this is polar and inside the cell is mostly water, and so you want the heads of the membrane facing in towards the cell. You want the fatty acid tails inside the plasma membrane, and then this layer facing the other way with the tails facing in, and they are lipid soluble, so they want to stay away from the water inside the cell and the water outside the cell. And then the phosphate region of the phospholipid here faces outside the cell. Any question about any of that? Now, the phospholipid is the main molecule in the plasma membrane, but there are other molecules in it, specifically carbohydrates and proteins. And we're going to talk a little bit more about the proteins in the plasma membrane. If the protein is only on one side of the molecule, like this one right here, then we call it a peripheral protein. And it can be on the inside, as shown here, or on the outside, as shown here. And that's what a peripheral protein is. A protein on a membrane, specifically the plasma membrane, but it can be any membrane, that is only on one side of the membrane, is called the peripheral protein. If, on the other hand, the molecule is inside the membrane, as well as maybe outside, then it's called either an integral protein or a transmembrane protein. A transmembrane protein is a protein that spans the entire membrane, and you can find it ending up on the other side of one side of the membrane and the other side of another the other side of the cell membrane. So transport proteins are transmembrane proteins. Any question about any of that? I think that's it for that slide. When we're talking about the plasma membrane, you should realize that it is described by a fluid mosaic model. So when we talk about animal cells, what's holding their cells together is a fluid, the cell membrane. And that's really striking. It always strikes me when I think about it. What's holding my cells together is nothing more than a fluid. Now it's true I've got the skin on the outside, which is dead cells, but what's holding my cells and tissue together is nothing more than this fluid. And how fluid is that fluid? Well, the membrane, the cell membrane, is as fluid as olive oil. So olive oil, if you look at it on your counter, there might be a little bit of it which is partially solid at the very bottom, but the majority of it is pretty thick fluid. And if you refrigerate olive oil, it'll become solid. But at room temperature, it is a fluid, maybe a thick fluid as opposed to corn oil, for example, but it is a fluid. Proteins move around on the membrane to function. We'll talk a little bit about that. Especially the proteins. If you take an A&P, you'll know that when a receptor binds to proteins, there'll be several receptors binding, and once they bind together, the proteins come together all in one region, and then usually the cell vaginates in that region. We won't talk about that now, but the point is that the proteins in the membrane move around, and that's true unless the protein is anchored either to something outside the cell, like the cytoskeleton outside the cell, or if the protein is anchored to the cytoskeleton inside the cell. If the protein is anchored, it doesn't move around. But if it is not anchored, the protein moves around in the cell membrane. And then the phospholipids also move around. Generally, they mostly just spin around, rotating. However, they can move, I don't know, like from this position to that position. And they just move by moving through. Any question about any of that? Make sure I discussed everything. The plasma membrane shows selective permeability. That means it allows the passage of some molecules, but does not allow the passage of other molecules. No large molecules can come across with the cell membrane unless they have a transport protein that will bring them into the cell. For example, DNA is a large molecule, and generally DNA does not get across the cell membrane. It's just too large to do that. If DNA is brought into the cell, then that's because there is a transport protein that will bind to the DNA and then bring the DNA inside the cell. Any questions about that? Let's come up to here. Membranes cannot cross the plasma membrane. Did I say that right? Ions cannot cross the plasma membrane. An ion is a molecule with a charge, and it does not like moving through this fatty acid region in a bilayer. So if ions cross the membrane, they do so only very slowly. Generally speaking, most ions do not move across the cell membrane. The exception is if there is a transport protein to move that ion across the cell membrane. For example, you might know if you eat a lot of potassium, potassium will bind to a transport protein and then be brought into the cell. Smaller molecules can readily diffuse across the cell membrane, and for our purposes that's mainly oxygen and carbon dioxide. But the oxygen outside the cell is generally higher than inside the cell, so oxygen will diffuse across the cell membrane. And then carbon dioxide, generally higher outside the cell than inside, so it will generally diffuse across the cell membrane. Water can slowly diffuse across the cell membrane. It doesn't like to cross the fatty acid region, but what happens with water is the fatty acids are moving around, and when one moves, water can sneak up and then wait for, on the other side, the phospholipid to move around, and then the water can move across the cell membrane. Generally speaking, that would be too slow for water to move into the cell like a plant in the desert, and so most cells will make a transport protein or something similar to that to bring water within the cell. Any question about any of that? The plasma membrane does have some unique function, especially in the bacteria or the prokaryotes. And the plasma membrane does have the enzyme for producing ATP. So all cells need to make ATP because that's the energy currency of cells. In eukaryotes, the majority of the ATP is made in the mitochondria of cells. Well, bacteria don't have mitochondria to make ATP, and so the bacteria make their ATP in the cell membrane of the bacteria. Any question about that? If cells are photosynthetic, meaning they can undergo photosynthesis, the photosynthetic pigments are found in the foldings of the bacteria cell. And we have a name for those. They're called chromatophores or thylakoids, similar to the thyroid. No, that's not right. What word am I trying to say here? Thylakoid, I think, of the chloroplast. So it's similar to the thylakoids of the chloroplast, but bacteria don't have chloroplasts, and so they engage in their photosynthesis by having what they need for photosynthesis put into the cell membrane, and then engaging in photosynthesis there. And generally there's a lot of that going on in the photosynthetic cells, so they have it more than just a normal cell membrane. They have foldings called chromatophores or thylakoids in the cell membrane in order to get their photosynthesis done. I'm trying to show you that here. Let me blow that up a little bit. And so that's part of the cell membrane, and it's just folded. Any question about any of that? Oops, didn't mean to do that. Let's see if I can get that back. And let's end the show here now. You can damage the plasma membrane, which will put a hole in it or burst it, and that can cause linkage of the cell contents. The cytoplasm will leak out once the plasma membrane is damaged, and that'll kill the cell, by the way. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and then the antibiotic polymyxin, like polymyxin B. It puts holes in bacteria plasma membrane, damaging the cells. Any question about any of that? All right. Let's talk about the movement of materials across membranes. We're going to talk about it as if it was the cell membrane, and that is the only membrane you see in bacterial cells. But please understand, this also applies to other membranes in a eukaryotic cell, such as the membrane of an organelle, like the endoplasmic reticulum. You can have movement of material across that membrane that's making up part of the endoplasmic reticulum. Any question about that? There are two processes that cells use to move movement across their membranes. The first is a passive process that doesn't cost the cell any energy, and the second is an active process that the cell has to use energy in the form of utilizing ATP. And what you're doing is you're moving things into or out of the cell by an active process, and that usually uses a transport protein. So there are three passive processes, and the movement of material in a passive process is always down the concentration gradient, meaning the thing moving into the cell has to be higher outside the cell than inside the cell, or it's not going to move into the cell by a passive process. There are three passive processes in cells: simple diffusion, such as oxygen moving into the cell; facilitated diffusion, and that's using a protein to move the molecule into or out of the cell. I'm going to use the example of the fructose transport protein. It acts by facilitated diffusion. When you eat fruit, the fructose in your gut will be higher inside the gut than inside the cell of the intestinal lining. And so fructose will move into the cell through the fructose transport protein by the laws of diffusion. So as long as fructose is higher outside the cell than inside the cell, then fructose will move into the cell by facilitated diffusion. Just simple diffusion and using a protein channel, or you call it a transport protein. This is costing the cell no energy because fructose is higher outside the cell than inside the cell. One disadvantage of this is once the fructose becomes equal outside the cell to that inside the cell, no more fructose, at least the net gain of fructose, will move into the cell. Any question about that? And then osmosis is the movement of water across the cell membrane. And osmosis is so important to living things that we give it a name, osmosis, and the water has to move by a passive process, meaning from higher regions to lower regions. So if the water is lower inside the cell than outside, water will move into the cell by osmosis. And water or osmosis requires the movement of water across a membrane. And when we're talking about the cell membrane, it will be the cell membrane. Water moving like from the soil into the root of the cell happens by osmosis. And then osmosis is also different because with simple diffusion and facilitated diffusion, the movement is a molecule which is a solute. Osmosis is a movement of a molecule of water which is a solvent. So osmosis is a little different. The solvent is moving. It's water moving across a membrane. And then it's limited to water. No other molecules use osmosis. And that's just by definition. All right, any question about the passive processes? We'll look at them in a little more detail in a little bit, but let's talk a little bit about the active process. Here, a solute molecule is moving from areas of low concentration to areas of high concentration. For example, if glucose were at low concentration in the gut and the cell had been, digesting food and breaking it down into glucose, there would be a lot of glucose inside the cell. Well, the cell is going to want to move glucose into the cell, even though it is at lower concentration outside the cell than inside the cell. So it will move the glucose into the cell by an active process, moving the glucose molecule against its concentration gradient. And how the glucose will move across the cell membrane is by a transport protein, and that will take energy because the molecule is not moving with diffusion. In fact, it's moving against diffusion. And so the movement of one glucose molecule into the cell will require the energy from 1 ATP to move that glucose molecule into the cell. And that's what an active transport is. Any question about the movement of materials across a membrane? So let's talk a little bit more about the passive processes, specifically simple diffusion, the movement of a solute from an area of high concentration to an area of low concentration. For example, if I had a beaker of water and I added a drop of dye, and there's a drop of dye settling in the water, eventually all of the molecules of dye would move and disperse in the water because the dye is soluble in the water. And that movement of the dye from regions of high concentration to regions of low concentration happened by simple diffusion. The dye moves from regions of high concentration to regions of low concentration by simple diffusion. And in cells, the two molecules that are important that move across the cell membrane by simple diffusion are the transport of oxygen and carbon dioxide. Now, simple diffusion can go across a membrane, like a cell membrane. But in this case, we're having simple diffusion happening in a beaker of water without any membrane being there. Any question about any of that? Well, passive processes are great, and simple diffusion is great, But what happens when the molecule can't move across the bilayer because of the fatty acid tails on the inside of the bilayer? How do you bring that molecule into the cell? Well, you use a transport protein that acts by facilitated diffusion. And like I said, the best example of this is the the fructose facilitated diffusion transport protein. Whenever you have fructose at higher concentration outside of the cell, the fructose can bind to the transport protein, and transporter and transport the same, and then move that molecule inside the cell. And because the molecule is moving from areas of high concentration to areas of low concentration, then we can move that into the cell without the expenditure of energy, because the molecule is moving by diffusion from areas of high concentration to areas of low concentration. And please try to remember this facilitated diffusion, oops, a little too far, in terms of fructose, because fructose works always as a facilitated diffusion. Once the fructose becomes lower outside the cell than inside the cell, then there will be no net gain of fructose into the cell. So fructose always works as a facilitated diffusion. This slide is showing you with glucose. And it is true that when glucose is higher outside the cell than inside the cell, glucose will move through the cell through a transport protein by facilitated diffusion. But once the glucose becomes lower outside the cell than inside the cell, this transport protein changes its shape and changes its function, and now it acts as an active transport protein, and it will move the glucose from areas of low concentration to areas of high concentration. So that's why the glucose example is not the best one to use as facilitated diffusion, because it at one time acts as facilitated diffusion, but at another time it acts as active process and moves glucose from areas of high concentration to areas of, it moves molecules from low concentration to molecules where they're at high concentration. All right, did I confuse anyone talking about glucose? Facilitated diffusion, remember, does not use ATP. Active transport always uses ATP to transport one molecule either into or out of the cell. In both simple and facilitated diffusion, the solutes are transported, and that transport happens down the concentration gradient, from regions of high concentration to regions of low concentration. With osmosis, we're not moving a solute, we're moving the solvent water across a selectively permeable membrane. And the water moves from areas of high water concentration to areas of low water concentration. Now there's a problem here, and that is when we're talking about things moving like water, you have to remember that we have it defined by tonicity, and tonicity is going to be the opposite of water, because water is the solvent and the solute is how we define tonicity. So if something is hypertonic, then it is more solute outside the cell than inside the cell. Let's make sure I've got that right. Yes. Okay. And when things are at high solute concentration outside the cell, that means they'll have low water concentration outside the cell. Any question about any of that? So when we're talking about passive transport like osmosis, it's important to remember that the cells can experience 3 different types of osmotic solutions. There's isotonic solutions, hypotonic solutions, and hypertonic solutions. Note that each of these is relative terms, so it's correct to say that the solution outside the cell was hypertonic relative to the solution inside the cell. Everyone clear on that? Isotonic is the easiest. It's when the concentration of solute molecules outside the cell is equal to the solute molecules inside the cell. So there's no net movement of water into or out of the cell. And the cell is happy because it's not going to be at any risk for drying out, water leaving the cell, or by osmotic lysis from water entering the cell. Let me see. Remember, the solute here is equal inside the cell and outside the cell, isotonic. That's how we define tonicity. But in this case, it would be the water that would be moving, and that would be the solvent. Hypotonic solution is when the concentration outside the cell is lower in solute concentration outside the cell than inside the cell. And that means there's a net movement of water into the cell. And that'll cause the cell to swell. The cell membrane will expand up until it reaches the cell wall. But if the cell wall is damaged by lysozyme or penicillin, there will be a hole or no cell wall, in which case the cell membrane will continue to expand and then will eventually burst, and then the cell will go under osmotic lysis, and that'll kill the cell. Most bacteria live in hypotonic solutions, and so that's why penicillin is a good antibiotic, and why it kills cells is actually it doesn't directly kill the cell, it just directly damages the cell wall, and then when water moves in, the water will kill the cell. Animal cells, of course, don't have a cell wall, and so if they are in an isotonic solution, water will move into the cell until the cell will burst. And then the last one, hypertonic solutions, the solute concentration outside the cell is higher than inside the cell. The water will then leave the cell, moving out of the cell, and that will cause shrinkage of the cell. The cytoplasm will decrease in size. The cell membrane will pull away from the cell wall, and then you might be able to see it, and that can cause plasmolysis, where the cell crenates, and that is the cell loses so much water that it can lead to death. And this is what happens when you have a bacteria cell falling in molasses. All right, any questions about tonicity? If not, I'm going to stop right here, and we will continue here in the next lesson. So I'll see you at 6:30 for the lab.